Hello everyone, my name's Ashlyn, and I'm just going to be introducing our paper that is Conversion of Natural Cytokine Receptors into Orthogonal Synthetic Biosensors by Haley, Eldesine, and Associates.
Synthetic receptors allow researchers to improve specificity and efficacy of targeted therapeutics. They are triggered by detecting certain cues. Orthogonal receptors work independently from innate signaling and are self-contained, recognizing the required signal mechanisms and not interfering with normal signaling. Synthetic orthogonal receptors can be incorporated for many functions, as they can be conditioned for certain outputs from specific cues. The authors used natural cytokine receptor ectodomains, which are parts of membrane proteins that reach to the extracellular space, to make MESA, a modular extracellular sensor architecture, receptors. This was done to produce natural ectodomain MESA receptors, which combine natural ligand recognition and synthetic outputs. Cytokines are produced by many cell types, expressing genes to regulate the immune system. They use a protein-based language that's crucial for communication, and cytokine language leads to limitations of synthetic receptors. These immune cells use their receptors to respond to signals in their own protein-based language. This impacts the same cell, the autocrine effect, or other cells in the organism, the paracrine effect. Limitations and mechanisms of synthetic receptors differ greatly. Communication with native signals and regulation without disrupting that native signaling can pose challenges, leading researchers to use synthetic receptors orthogonal to endogenous signaling.
Hi, I'm James, and we're going to be talking about the research design. So, the main goal of this research is to create a novel receptor that acts orthogonally to other receptor pathways in the cell. So, we've seen things like this with synthetic factors and receptors. One specifically is called MESA that they end up using in this study. And they're very customizable because they can use things like antibodies or very specific ligands, but we haven't really seen soluble ligands that are easy to produce. So the method in which the researchers go about solving this problem is by kind of mixing and matching receptor structures of pre-existing cytokine receptors and basically customizing the MESA receptor to bind to soluble ligands. And let's see how they go about doing that. So the first thing that the researchers had to do here was that they had to find currently existing cellular receptors that would be a good fit to couple with the mechanism of the MESA receptor, which is the fully synthetic receptor. And the ones that they found that would be a good fit, or the ones that they hypothesized would be a good fit, are VEGF, IR10, TNF, which is the tumor necrosis factor receptor, and TGF. And they chose to start with VEGF first because the mechanism in which it works is very similar to that of the MESA receptor, in that it dimerizes and can bind to a ligand separately. But when it binds to the ligand, that is the only time when it will release the transcription factor. And additionally, the VEGF receptor was also shown to work in a similar mechanism from other researchers. So they chose to move forward with VEGF, and they wanted to see if changing parts of the sequence would affect the expression of VEGF on the outside of the cell. So when doing this, they found that a change in the signal sequence did not actually affect the expression on the outside of the cell, and neither did the transmembrane domain. So the extracellular domain was the most important part in keeping the expression of VEGF on the outside of the cell. So using this, they created a configuration that was able to respond very well with the plasmid expression of the ligand. So when there was a lot of ligand in the cell, the receptor also showed expression on a reporter gene. But when they tested this with a exogenous ligand, as in only ligand in the solution, it didn't actually have as high as a signal. So they chose to experiment with the other receptor options they had. So when testing IL as a receptor, they found that it responded very well to ligand on the outside of the cell. And they tested different configurations using IL-10, along with the intermembrane domain portions of the mesoreceptor. And they found a very specific combination that worked very well and had the best expression of the reporter gene. Then, after this, they wanted to test how would they integrate this receptor into the bacterial genome. And they did this using transposons, but they wanted to see if expressing the ligand separately from the receptor would have any effect. So they tried adding the receptor alongside the ligand on a single transposon, and then adding them separately with different ones. And they ended up finding that when they were on the same transposon, i.e. when the gene for the receptor and the gene for the reporter were very close to each other, or the gene for the ligand were very close to each other, there was the best rate of signaling and expression of the reporter gene. So when they found this out, they wanted to see if they could use this. So they expressed the new receptor that they made with the new configuration that they found previously and used it alongside the CAR receptor in order to track the level of expression, making an AND gate with it, basically. And they found that the cells that were tested with the exogenous ligand and the ligand for the CAR receptor responded very well, even including with ligands expressed by other nearby cells, which was very impressive. So now that the receptors were shown to work with exogenous ligand, they wanted to test these receptors in a practical application. So the first thing they did was that they expressed the IL-10 receptor in a T cell, and they used a transcription factor that increases the cytotoxic activity in the T cell. And they showed that this worked and that the transformed T cells were able to sense the IL-10 in solution with a threshold of 32 nanograms per milliliter. But after sorting for cells that had the most transposons, the threshold was actually lowered to 2 nanograms per milliliter, showing that an increase in the expression of the receptor and the ligand would be able to lower the sensitivity of signaling. And in addition to this, they wanted to test genetic logic circuits using these modified receptors. So they used the VEGF receptor and the IL-10 receptor in combination to create an OR gate and 2 different kinds of AND gates. The OR gate worked by having the same transcription factor on both receptors, so that when either or of them were activated, you would have expression of the reporter. And they did 2 different ways of making an AND gate. The first one worked by having the 2 transcription factors dimerize to activate the expression of the reporter gene, and the second one worked by having these 2 different transcription factors activate the expression of a dimer transcription factor that would activate expression of the reporter gene. And this did show some promise, as the observed behavior showed some resemblance to the desired behavior, but there was some leakiness in that there was expression of the transcription factor for the reporter gene when there was not intended to be.
Hello, my name is Katan Patel, and from the earlier slides, we know that the research are out to determine whether natural cytokine receptors can be converted into orthogonal synthetic biosensors using the MESA system. So starting with the VEGFR family and its conversion success, we know that VEGFR1 and 2 are both mesoreceptors that showed a strong ligand dependency for activation. This just means that when exposed to VEGF, the receptor showed a larger response. And it's good to note that the natural transmembrane domain did show improved performance compared to the synthetic ones. The I10R receptors and their conversion success, so IL-10R alpha and beta designs were stably expressed and strongly inducible with IL-10 concentrations as low as 16 nanograms per milliliter, which shows its validity with physiological limits. It also showed that it has functionality when tested in both two different cell lines. The TNFR and TBF beta R challenges, the conversion between these two have led to poor inducibility or unstable expressions, which kind of shows you the limits between natural ECDs and synthetic intracellular domains. All right, this is the application and integration of the findings. So you had the successful conversion of VEGFR and IL-10R receptors, which shows you that the natural receptor ectodomes can serve as modular synthetic biosensors, specifically with orthogonal signaling. So by demonstrating that the IL-10 and message receptors could be used to reprogram T cells. So when exposed to IL-10, the engineered T cells activated an anti-tumor gene circuit, such as CARs, which is chimeric antigen receptors. This transforms IL-10 from a tumor protective signal into a trigger for immune activation using the AND gate, which using the AND gate reduces off-target toxicity of the CAR therapy. So overall, what this means is that the receptors, that once it's limited to the natural signaling, can be rewired to perform orthogonal functions and provide more precise therapy that responds only to the disease-specific cytokine environments. Hey, this is Katan Patel again. I just wanted to expand slightly more on the limitations. So, some of these incomplete receptors, when folded for some constructs, it showed that the ECD orientation is critical for this ligand receptor to work. So, the conversions of TNFR and TBGF beta R did not show strong ligand induced signaling, which is mostly due to a orientation or geometric issue. There was cross reactivity with native receptors that could occur in cells, which shows that when the ligand was absent, several receptors still showed some basal activation, which essentially means some leaky channels. And additionally, these ligands require a more optimization. So even though the ligand dependent activation showed a signal, the intensity varied depending on the receptor family. And then the experiment was done on 2 different cell lines, but it has not been done yet in vivo, which it still needs to prove its validity for in using human trials.